Well, good morning, everyone. For those of you that are interested in ecological speciation, sorry, but my talk is not focused on this topic. But I promise that I will show you some examples about how ecological factors are driving or may drive some adaptive processes on insular populations of Salamandra. And these processes involve a reproductive and also a behavioural shift in two small island populations. So before explaining these experiments or these studies, I want you to show or to give you some relevant information on the species. So I don't need to be close to this microphone, right? Okay. So Salamandra, the fire salamander, is a widespread species going from southern Iberia to eastern Europe. Well, it shows a very high morphologic diversity. So, for instance, here I'm just showing 6 different pictures that all of them are coming from the Iberian Peninsula. Well, from this morphological diversity, people started to describe subspecies. So right now there are 16 subspecies described, and 10 of which are in the Iberian Peninsula. But the most important thing here in this species is that it's showing reproductive, well, interspecific reproductive modes. So in salamanders, there are only 2 species that show interspecific diversity, and it's this one, Salamandra, and also Salamandra algira, the sister species here in Morocco. As I say, there are 2 reproductive modes. This is a female about to give birth, and depending on the strategy, it can produce many of these small larvae that they are free. Well, they are water dependent, so they have to stay in the water for at least a month. And this is called this strategy is called ovoviviparity. This is the common strategy in this species. And then we have viviparity here, where the female is just producing or is laying a few large and fully metamorphosed juveniles. So they are completely independent to water. As I said, in the Iberian Peninsula, there are 10 subspecies described. Probably most of them, they are not valid because genetically there are some admixture. But what I want to show you here is that the viviparous subspecies is this one, the Salamandra bernardesi, and is covering this part of the northern Iberian range. And also we have Salamandra fastuosa here, light blue, that there is some admixture. There are ovoviviparous and viviparous populations. And then we have this subspecies, the red one, in which we found 2 island populations right here that there are also viviparous. So 10 years ago, this was in an evolution paper, Mario Garcia Paris and co-authors were interested in studying the evolutionary history of the species, but also they were trying to identify the origin of viviparity in this species. So what they did, it was they analyzed the genetic relationships among northern Iberian populations using mitochondrial markers and also nuclear markers. So what they found here is at the mitochondrial level, they found 3 divergent clades, 2 of which are represented here in the northern Iberian Peninsula. So what they suggested was that in the late Pliocene or early Pleistocene, by vicariant processes, these populations were isolated here. So these bricks represent like barriers, in this case glacial barriers probably. And then afterwards there was a spread from here, from this mitochondrial clade, through the coast. It was eastern colonization to the east. So what they suggested was that the viviparity occurred here, and then this viviparity condition, but also the stripe coloration here that you can see, it was moved from this population, from this region, to the other, well, to the Pyrenees. They didn't spread southern or western because of the mountains that are covering this portion of the northern Iberian Peninsula. But in that study, they did include, as I said, the 2 islands right here. So we don't know the origin of those islands. Also, another interesting study to understand the differences between these reproductive modes are this one from Buckley in 2007. So what it's showing here is the ontogenetic processes, the differences between the development of embryos for the ovoviviparous salamanders and for the viviparous salamanders. So what we have here is a temporal axis from the fertilization to the metamorphosis, and comparing the main difference between the two reproductive modes. So there are many differences between these two strategies, but I want you to focus just in these three main differences, and is that the time that they hatch, the time that they start the active feeding, and also the metamorphosis. So if we compare the two reproductive modes, we see that for the viviparous salamanders, that is here after 9 days, the embryos start to hatch, and also the start of the active feeding is just after a couple of weeks, and the metamorphosis occurred after 3 months. So the salamander, the females, just lay these terrestrial juveniles. But on the other side, we have the hatching, the active feeding, and the metamorphosis occurring, well, 90 days, and the metamorphosis occurring after a month or maybe 2 months when the larvae are in the water. So there is a clear acceleration of the development of viviparous embryos. So the two islands I'm studying belong to the Atlantic Island National Park on the northwestern Atlantic coast. And this national park is formed by like 8 main islands and 20 islets. These islands are young. The oldest islands are about 8,000 years old, because they were basically mountain hilltops on the continental area that were completely isolated after the last glacial maximum when the sea level rise. So these areas got completely isolated from the mainland. Another characteristic is that they are very close to the mainland. It's only 2-3 kilometers, and they are small. The whole national park is about 84 square kilometers. So these are the two islands. The two island population right now is in the Ons archipelago. We have here this main island. There is a huge population of salamandra, and also in this archipelago, the Thies archipelago, we have a population right here. It was here before, like 10 years, 20 years ago, but is now extinct. So the first point that we were raising up what is the origin of these island populations? It's a new origin, or is the result of an ancestral colonization from these viviparous populations until these island populations? So what we did, it was a simple approach. It was using mitochondrial markers. We sequenced the samples from viviparous populations on the islands, but also ovoviviparous from the mainland, and also covering some viviparous and ovoviviparous populations across northern Iberian Peninsula. So the idea is, if this was an ancestral colonization, we would expect these two island populations closely related to these ones here. What we found is 2 different clades, well supported. All of the viviparous, the pure viviparous populations were here in this clade. And then we found another clade with the ovoviviparous populations, but also with the 2 island populations. So this is suggesting that the origin of these island populations is independent. I mean, the viviparity in these island populations are independent. But of course, mitochondria is not the best approach to see if this colonization was recent or not. So we went to a more population genetic approach. And in this case, we sampled. The sampling design here was trying to get, we got actually more than 30 adults per locality. So we covered the 2 island populations here with these red asterisks, and also 3 coastal populations and 2 interior populations. So we genotyped some microsatellite markers, and we assessed genetic structure, genetic diversity patterns, demographics. So again, if these populations were the result of a recent colonization process, we would expect some admixture, some genetic admixture between these populations, right? This is what we got. The genetic structure was very clear. From the 7 populations, we got 6 genetic dims represented by different colors. So each of these island populations constitutes a different genetic dim. But also what was striking also was that these coastal populations also represented different genetic demes, while the two interior populations were connected. So this probably is because these three peninsulas here, especially this one, represent historical islands. So probably these two island populations and these three historical islands were suffering the same processes. Also, we studied the genetic diversity values, historical population sizes, and historical population growth. And what we found, it was like similar values for the two island populations, and for the three historical islands, we found more or less similar patterns. So it was higher genetic diversity for the interior, higher population size, and higher population growth for the inland populations. And we didn't find any evidence of migration. So the conclusions on this topic is that based on the differentiation at the mitochondrial level and also on the differentiation at the microsat level, we suggested that the viviparity here occurred independently, and it was rapid. I mean, rapid in terms of comparing with the northern Cantabrian mountains. This probably happened during the last few thousand years. So what factors led to this evolutionary change? One could be the genetic isolation and drift, but we think that this is unlikely because, as I said, the two island populations and the historical populations are showing similar genetic patterns. So it's unlikely to think that this viviparity occurred by drift, just by drift alone. So phenotypic plasticity. Also, this could be explaining why these animals are viviparous, but we also think that this is unlikely. First, because we also were bringing females from the islands to the lab, and we kept them in captivity with water availability, waiting to see if they are producing larvae or they are producing juveniles. All of them produce juveniles. So it seems that depending on whether or not they have water in the big containers, they are just producing terrestrial individuals. And also because the study I showed before, that they are this differentiation of the embryos, it's pointing out that there is something fixed here. And the third possibility is the ecological factors. This could be quite possible. Actually, the lack of water and low precipitation would force salamanders to struggle and to adapt to dry conditions. And actually, these islands are drier comparing to the mainland, and also one of the islands there is lack of water. This lack of water is because a few years ago, or 2 decades ago, there was eucalyptus plantation on the island, so the water just is now desiccated because of this plantation. But this is very recent. So this could be one possibility, and one obvious possibility. The problem here is that it's kind of tricky, because one of the islands, the ONS island, the large island, has also small ponds where other two species, an anuran and anut, are breeding there. So if these animals can breed there, why not Salamandra? So I'm going to stop here on this topic, because I could just keep talking on this, but this is just speculations. We don't have data to see if this is the right cause or not. So now I'm changing to the second topic. I'm trying to be fast, sorry. And this is also very interesting. As all of you probably know, or many of you know, Salamandra is a nocturnal species. It's very common or very easy to find them with the right temperature and humidity levels at night. But there is no activity during the day. Sometimes in foggy days with the sunrise or sunset, is it possible to see them active, but that's it. So in the ONS population, the large population, every day we went there, no activity during the day, and at night, dozens or even hundreds. Actually, once I found 700 animals in this small island. But this was striking when we went to the other island since 2003, and we started to see some activity shift. So in this graph, I'm just showing the number of active salamanders in blue during the day and in red during the night. So this is quite striking. I mean, every time that we go to the islands, we go to the perfect time when there is rain, when there is high humidity, and with the temperature when the temperature is okay. So we started to go there, and we started to see this pattern. So during the day, we found a few animals, and then at night, no activity. Again. There was only one night 2 years ago where we found an explosion, and that explosion was at night. But I will come to this later. So we also were measuring the physical condition of these mainland, coastal, and island populations, and we found that this diurnal population showed the lowest physical condition, which could be related to the diurnal activity and also could be related to a low fitness. So what drives this behavioral change? Maybe predators, maybe a high predation pressure on this small population could drive to this behavioral change, right? We have predators. We have predators on this island. We found several salamander carcasses on the island, and all of them were eating like this, were half eating. The predator is eating the head and the forelimbs, but discarding the tail and the hind limbs. So potential native predators all of them are diurnal. We have the common buzzard, the goshawk, so there are 2 birds, diurnal, and also we have a snake, also diurnal. So these predators don't look like could be the right predators to change this behavior, right? But then we have 2 invasive mammals. There is an old and huge population of ratus, of black rat on this island. This population is huge, especially in summer. But every night or every day we went to the islands. It's very easy to see them, and actually they are trying to steal some food from us. So it's considerable. I don't have the numbers, but it's huge. And this is all, well, we consider this is all because probably they colonized the island with sailors 1,000 years ago. And on the other hand, we have the American mink. It's recent and rare. Recent because in Galicia we have 4 farms for many years. Intentionally or accidentally, people were releasing animals from these 4 farms in Galicia. And some of them managed to cross the seawater barrier and colonize many of these islands on the national park. Right now, I think there are like 3-4 animals on the island, so could be a potential predator, but I mean, there are very few animals. So what we did here was an experiment with plasticine models. This is a plasticine model, and this is the living one. So this is very cool to work on this system because any attack, birds, mammals, or whatever, leave very easy marks to recognize afterwards. So the idea was, well, I produced 60 of these salamander models, and I spread across the island. And I was checking these models with the sunrise and with the sunset to see what predators are attacking, but also to see if these predators are diurnal or nocturnal. So what we got is that all of the models that were attacked, all of them were having this very easy to recognize rodent incisor marks. So you see here on the head. All of them were attacked by rats. And on San Martino Island, about 10%, well, from 10% to 30% were attacked only at night. None of them were attacked during the day. Of course, mammals have this very sensitive smell, so it's possible that the rats are just biting. But on the other island, on the islands, the number of the number of plasticine models were attacked only less than 10%, I would say. But this is still an ongoing study, so we are still generating data. We are also using camera trapping. So with this camera trapping, we know the predators that they are on the island, but we also want to know if there are more predators. So with this camera trapping, we are also checking what potential predators are on the island and at what time they they are occurring, right? So these cameras are very sensitive. We can detect birds. We can also detect some small lizards right here. We can detect mammals like this rabbit, but 80% or 90% of the pictures and videos were detecting rats. And also we have recorded with pictures and videos some rats attacking the plasticine models. So I'm going back again to that night where we saw that explosion of salamanders. So yes, we were like very surprised because after a few years sampling this island, that night was, so what the hell is going on here? I mean, there are so many salamanders right now. That night was a very rainy night. So I was like speculating whether or not the rats could be active that night. So luckily we have data for, well, climatic data from the climatic station of this island for every 10 minutes. So I could compare the number of active salamanders here on red with the temperature, precipitation, and humidity. So if you see the temperature or the precipitation are maintained quite stable here. Temperature about 10 to 14 degrees, and humidity from 60 to 90. It's kind of stable, and there is no pattern. But if we look at the precipitation levels, that night, that was a very heavy, rainy night, you see the correspondence here. Of course, this is just one point. We are still generating data, and we are willing to go to the islands more often to get more data. So as I said again, nocturnal activity was linked to the heavy rain, and the absence of mammals could be related to this pattern. Just the conclusions. So yes, the salamanders look diurnal, although there are some nocturnal activity. These salamanders have low physical condition that could be related to this diurnal activity. So answering to the question that I presented, salamanders do have predators. The models suggest that the only predator is the black rat, and the camera trapping so far didn't detect any other predator. Of course, further work is needed. So what I'm doing now is starting a collaboration with the zoo at Vigo in Galicia, where I want to bring salamanders from the two islands and to put them in isolated big plastic containers to see, well, to detect or to track the activity of these animals. This was done in collaboration with Adolfo Cordero from Universidad de Vigo, and we want to thank Maria Casal and the staff from the National Park for their help during these years. And I think I didn't show this picture before. This is the San Martino Island, from another island of the Sears archipelago. Thank you. Thank you for the.
Thank you.